Hi, my name is Ziwei. Our group is Group 4. Our title is The Role of Chromatin Remodeling of MHC Class 2 Gene. Introduction. MHC class 2 gene is a class of major histocompatibility complex molecules. It found only on the professional antigen-presenting cells. These cells are important in initiating the immune response. The MHC class 2 gene can use to present the antigens that are extracellular or exogenous. There are three classical isotypes of MHC protein complex, which are also known as the human leukocyte antigens. They are the HLA-DR, HLA-DP, HLA-DQ. Chromatin remodeling is the rearrangement of chromatin from a condensed state to a transcriptionally accessible state to allow transcription factors or other DNA binding proteins to access DNA and control the gene expression. The role of chromatin remodeling is to establish the specific gene expression patterns and to maintain the transcriptional state in eukaryotes.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Amir Shafiq bin Musli Kamal. My matriculation is SB19042. So today I will talk about the current status. So in recent years, researchers have discovered a great deal about chromatin remodeling, including the roles that different protein complex, histone variant, and biochemical modification plays in this process. However, a great deal remains to be learned before chromatin remodeling is fully understood. So the idea of this research paper is it apply MHC class 2 locus in coordination of long-range coordination of MHC class 2 gene expression. It also apply histone acetylation by using transcription of active MHC class 2 at multiple levels. It also regulate MHC class 2 gene expression using a long-range chromatin remodeling. So this research is based on the focus of the role of chromatin remodeling, which is particularly remodeling that involve histone acetylation in the constitutive and indivisible regulation of MHC class 2 gene expression. The clinical trial that involved in this research is HDAC inhibitor alone or in combination with other agents for treating various tumors are currently in preclinical or clinical phase 1 and 2. So the novel therapeutic modalities that can modify MHC class 2 genes at the epigenetic level are emerging and currently in clinical trial and preclinical trials. The commercialization of this research in genetic engineering is it used measure histocompatibility class 2 gene in expression, which is a new finding as it was used in immunotechnology of immune response development and the technique of histone acetylation in DNA. So the chromosome study of opening the chromatin in a new way in a DNA studies. I'm SB19042. Thank you.
Hi, I am Ali Rajai bin Baharudin. On today's topic, the role of chromatin remodeling for MHC class 2 gene. Specifically, I will be talking about genes and proteins involved and the significance of this study. So let's go with genes and proteins involved. Histone tail modification. Histones are DNA strands wrapped around an octamer of core histones to form the basic chromatin unit, the nucleosome core particle. This can be visualized in diagram 1. As you can see from diagram 1, the thread-like structure or the hair-like structure in the middle is the DNA. The DNA will coil around the histones. The histones are the blue-colored cylinders. So once it is coiled around histones, it is called a nucleosome. Other than that, the other string-like structure outside the histones with the black colored strings, those are histone tails, which is very important to understand, as I will explain later. And these histone tails can be modified through acetylation, phosphorylation, methylation, ubiquitination, and citrullination. Altering these histone tails will alter the chromatin between being folded and unfolded conformation. This results in either the segment of the chromatin being able to be translated or not, thus either activation or deactivation of genes. So this is histone tail modification. Now, there are an example of enzymes that can be used to alter these tails, such as histone acetyltransferase or HAT, and histone deacetylase HDAC. So one is it acetylate, the other one deacetylase, which catalyzes histones acetylation and deacetylation, proximal promoters of the MHC class 2 gene. So that one is histone tail modification. That is one of the example where one can alter the chromatin structure, which is in a way a post-translational modification by activating or deactivating a certain part of the gene. In this case, the MHC class 2. So furthermore, proximal promoters of MHC class 2 genes. Proximal promoters are promoters that is located about 250 base pairs upstream from the transcriptional start site. This means promoters are a bunch of codes that promotes transcriptional factors to bind onto it, or in a way to start any transcription processes. So proximal promoters are promoters that is located about 250 base pair upstream from the TSS. The proximal promoter SY specifically, the SY module consists of multiple promoters such as X1, X2 and Y box elements with Tata box. This promoters plays an important role in chromatin remodeling, which alter MHC class 2 genes. This SY module regulates whether the MHC class 2 gene will be able to be translated or not. So these are two examples of factors that can affect the chromatin modeling and ultimately affect the MHC class 2 expression, gene expression. So let's go to the significance of this study. One example is mycobacterial infection. One such example of mycobacteria is Mycobacterium tuberculosis, which are known to be really hard at curing the infection or getting rid of such infection from human system. Through a study of mycobacteria infected macrophage, one of the reasons why Mycobacterium tuberculosis or Mycobacterium itself is really hard to cure is because they tend to shut down the upregulation of MHC class 2 gene. So one example is, through a study on Mycobacteria infected macrophage, the macrophage seems to be reluctant to express MHC class 2 gene upon IFNY gamma stimulation. IFN gamma stimulation is a molecule or cytokine released to activate white blood cells, in this case macrophage, or it can activate other white blood cells to like killer T cells, etc. Further studies shows that these bacterium significantly inhibit the IFN gamma induced histone acetylation in the HLA-DRA proximal promoter. This is important in understanding the mechanism of post-translational modification, specifically for chromatin remodeling. Now it says over there induced histone acetylation. This means it inhibits the histone from being opened up, acetylation. Deacetylation is doing the opposite, so it closes the histone together, so the chromatin is packed. Once it is packed, translations is not possible. So in a way, this bacteria blocks the MHC class 2 from being translated. This is important because it opens new areas of understanding which may lead to more discoveries regarding the possible mechanism and technique that can be used to alter the DNA expression for MHC class 2. Lastly, this is another important significance of this study. Anti-tumor therapy. Tumor cells such as dendritic cancer cells from cancer patients shows MHC class 2 expression being silenced. and some does not express MAC class 2 upregulation even though IF and gamma were being introduced. Now again, IF and gamma is cytokine that is used to activate cells like macrophage, etc. In recent studies, TSA treated tumor, TSA, a drug used to treat tumor cells, manifests an effective tumor cell vaccine in vivo. TSA induces cell surface expression of MAC class 2. These are some of the examples of understanding the chromatin modeling importance. That is all from me. Thank you.